Good afternoon, class. So today is going to be chapter 3, part 2, online lecture. And we're going to pick up where we left off talking about diffusion, okay? So remember, diffusion is when particles move from where they are highly concentrated to where they are lowly concentrated. And diffusion does not require any energy, okay? That's called moving down the concentration gradient from high to low. All right, doesn't require any energy. All right, and a special type of diffusion is osmosis, which is the diffusion of water moving from where it's highly concentrated to where it's low concentrated. Okay, and for a cell, okay, it has to limit how much water can enter, or the cell can swell and essentially lyse, or it can even shrink. So depending on what type of solution a cell is exposed to, and we're going to talk about the different types of solutions, isotonic versus hypertonic versus hypotonic. Okay, a cell can stay the same, it can shrink or swell, okay, depending on what type of solution it's exposed to. All right, so tonicity is the ability of a solution to change the shape or tone of cells by altering the cell's internal water volume. All right, so isotonic solutions have the same osmolarity, essentially the same concentration of solutes. So the inside of the cell has the same concentration of solutes as the outside of the cell, which is this isotonic solution. They have the same concentration of solutes, okay? So the volume remains unchanged, meaning there's no driving force for the water to move in or out of the cell because it's already at equilibrium. Once the concentration of the particles are the same inside and outside of the cell, there's no driving force for the water to move, all right? So essentially the cell's at equilibrium, okay? Hypertonic solution has higher osmolarity than inside the cell. So essentially, you have higher concentration of particles. I just usually just say salts because it's just a common thing. So hypertonic solution, you have a higher salt concentration outside of the cell. I'm sorry. Yeah, you have a higher concentration of salt outside of the cell. All right. So the way I remember it, okay, and again, this isn't the scientific meaning, but it just keeps it straight in my head, okay. So when you have, so I always say, okay, hypertonic, okay. So I always say hyper. That prefix hyper means high, okay. So, and then tonic doesn't mean salt. I know that's not the definition of tonic, but I always know you're talking about the particles. So I always say high salt, okay. So hypertonic solution has high salt, therefore, and this is in relation to the cell, okay. So you're always talking about the solution in relationship to the cell, the inside of the cell, okay. So the hypertonic solution has high salt. So remember, when a solution has high salt, the water concentration is the opposite. So if it has high salt, that means it has low water, all right, compared to the inside of the cell, which is going to have high water on the inside of the cell. So remember, water always moves from high to low. So water is going to move out of the cell, okay, and the cell is going to shrink, all right. So again, hypertonic solution means high salt solution, okay. If you have high salt solution, that means you have low water, okay, in the solution. So that means inside of the cell has higher concentration of water. So water always flows from high to low. So it's going to flow from inside of the cell out, and the cell is going to shrink, okay. Hypotonic solution, the prefix hypo means low, okay. So low salt solution, A low salt solution is going to have a high water concentration, okay? And that's in relationship to comparison to the cell. So high water on the outside of the cell, high water solution, that means water is going to go from outside of the cell to the inside of the cell, and the cell is going to swell, okay? And if the cell swells too much, it can burst, okay? And that's referred to as lysing, all right? So again, in isotonic, iso, the prefix iso means same, okay? So isotonic, same salt solution inside and outside of the cell. So there's no driving force for the water to move, okay? So again, hypertonic solution, high salt solution, meaning it has low water in the solution, okay? Therefore, inside of the cell has higher concentration of water, so the water flows from inside out, and the cell shrinks. Hypotonic, low salt solution, meaning it has high water in the solution, okay? And the water always flows from osmosis from high to low. So it's going to flow from the solution to inside of the cell, and the cell is going to swell. All right, so you just remember it that way, and you keep it straight in your head, okay? But always remember that the solution is in relationship to the cell, okay? So just imagine you're dropping a cell in these solutions, okay? Just imagine it that way. A simple experiment that I used to do is dealing with grapes. I would do isotonic solution. I would do a solution of grape juice. So I would drop a grape in the grape juice. So that would be my isotonic solution, and the grape should stay the same size. For the hypertonic solution, I would just use really concentrated sugar water. Okay, so I would drop a grape into a really high concentration of sugar water. Okay, so the grape should shrink in that sugar water. And then a hypotonic solution, you just drop a grape in just water. Okay, water with no sugar in it. That's going to be a hypertonic solution, and the grape should swell. Okay, and again, that's just a simple experiment I would do in biology class, but it will kind of let the students see it and bring home the point. So again, just imagine dropping a cell in these solutions, okay? And then you have to think about in your mind, what would the cell do in these solution? And again, you want to always, what you're really trying to think about is how the water is flowing, okay? So remember, water is always flowing from high to low, but you got to think about the solution, whether it's hypertonic or hypotonic, because a hypertonic solution has high salt, okay, high salt, therefore it's going to have low water in the solution, therefore the cell's going to have more water inside of it, and the concentration of the water in the cell is going to be higher, I should say it that way, and water is going to flow from high to low, from inside the cell to outside where the solution is, and the cell's going to shrink, okay? So always think of, you're thinking in terms of, how many particles, whether the particles are high or low in the solution, but I'm only doing that because I want to figure out where the water concentration is high and low, whether it's high in the solution or inside the cell, or low in the solution or low inside the cell, so I can see how the water flows, okay? Because the water is always going to flow from high to low. All right, so this, I'm sorry, I didn't mean to skip that slide. So let's go back to this illustration. So again, isotonic, iso means same salt. Okay, so same salt, the solution has the same amount of salt in the solution as inside of the cell. Therefore, there's no driving force for the water to move. Okay, so isotonic solution, the red blood cell stays the same. It doesn't change. Okay. Hypertonic solution, hyper, that prefix hyper means high, high salt, okay, high salt, and you're going to have low water. When you have high salt, that means you have to have low water. It's always, those two are always the opposite. So high salt means you have a low water solution, therefore you have higher water concentration inside of the cell, so water flows from high to low, so water is going to flow out of the cell, and the cell is going to shrink. Hypotonic solution, low salt, that means you have high water, Okay, so inside of the cell is low water, so the water is going to flow from the solution inside of the cell, from the solution inside of the cell. Therefore, the cell's going to swell. Okay, so this is just a image of a red blood cell and three different types of solutions. Okay. All right. So again, let's talk about active transport. So we've been talking about passive transport. Now let's talk about active transport. So we again with active transport. Okay, active transport is going to require energy. The word active means that it's going to require energy. Okay, and there's two major types of active transport. All right, you have active transport and you have vesicular transport. Okay, both require ATP. All right, to move solids across the plasma membrane. For any of these reasons, either the solute is too large for channels, or the solute is not lipid soluble, or one of the most common reasons is the solute is not able to move down a concentration gradient, meaning it's moving up, meaning it's going, instead of going from high to low concentration, it's going from low to high. And you would think about, well, why do you want to go from low to high? It's because certain things you want to, a lot of times the cell wants to concentrate certain things inside of the cell. So a lot of times it has to do this by going against the concentration gradient. So it has to go from low to high. And that requires energy, ATP, okay? And when the solutes or the particles are too large, this is where you're gonna get into exocytosis and endocytosis, okay? So those exocytosis and endocytosis, those are active transport processes, and we'll talk about those also. But that's gonna be dealing with things that are too large or not soluble to pass through the plasma membrane. All right, so active transport requires carrier proteins, and again, these carrier proteins are specific, okay? So they bind very, very specific with the substance that's going to be moved, all right? And they move against the concentration gradient. That's very, very important. So they're moving from low, where it's low concentrated, to where it's high. All right. So again, in order to move against the concentration gradient, sometimes they use the word going up the concentration gradient, as opposed to going down concentration gradient. So remember, going up concentration gradient, you're going to require energy. Okay, going from where something is low concentrated to where it's high, that requires energy. Example of active transport, one of the main examples that they use is the sodium potassium pump. Okay, is very important for maintaining the electrochemical gradients that are going to be the driving force of your action potentials, which we'll talk about when you get to the nervous system unit. But this sodium potassium pump is going to be very important for maintaining electrochemical gradient that's going to drive action potential in muscle and nerve cells. Okay, but so essentially what you're doing, what the cell's doing with this sodium potassium pump, is pumping three sodium out of the cell and 2 potassium in. So essentially it's concentrating sodium out of the cell, okay, and then it's pumping 2 potassium in. All right, so it's concentrating sodium out and concentrating potassium in. All right, and what happens is you end up with an overall net negative charge inside of the cell when it's at a resting membrane potential. Overall net negative inside the cell, and overall net positive outside the cell. Okay, so you have that electrochemical gradient, negative inside of the cell, positive outside of the cell. What happens is during an action potential, all right, that sodium, we have sodium gated voltage gated channels that open up during action potential. And what's going to happen is if you have a negative charge inside of the cell, obviously those sodium that's concentrated outside the cell are going to rush in the cell, and it's going to make this inside of the cell become more positive. Okay, and again, that's beginning step of depolarization for an action potential. And then we'll talk about action potential in detail when we get to the nervous system. But without this electrochemical gradient that sodium potassium pump creates, you wouldn't be able to have action potentials. That's what I'm trying to get home here, is that the sodium potassium pump is very, very important. Okay, and it creates this electrochemical gradient, that's essentially concentrating sodium outside of the cell and potassium inside of the cell, and it's creating this negative electrical charge inside of the cell, overall charge inside of the cell, and a positive overall charge outside of the cell. Okay, and that's the driving force for action potentials when those occur. Okay, so again, very important, and this requires energy. It's one of the most studied pumps. Okay, and it requires ATP. All right, so this is showing you a schematic of the steps of the sodium potassium pump. So you get right here, you get 3 cytoplasmic sodium bind to the pump. So you get 3 sodium bind to the pump, and then the sodium binding promotes hydrolysis of ATP. So cleaving the ATP, using it for energy. Energy released during this reaction phosphorylates the pump. Step 3, the phosphorylation causes the pump to change shape, and it's going to expel the free sodium ions outside the cell. And then step 4, 2 extracellular potassium bind to the pump. Step 5, potassium binding triggers release of the phosphate. The dephosphorylated pump resumes its original conformation. And then step 6, the pump protein binds ATP, releases potassium to the inside, and sodium sites are ready to bind sodium again, and the cycle repeats. Okay. All right, so we talked about the sodium potassium pump was one of the most common examples of active transport. Okay, another example of active transport is vesicular transport. So this is when you are doing endocytosis or exocytosis, okay, or transcytosis. Okay, so the name tells you just what they're doing. Okay, endocytosis. Think of the prefix "in." Endo sounds like in. Okay, in. So you're essentially transporting things inside the cell. Okay, so you have different types of endocytosis, and they are just really depending on how large the particle that you are bringing inside the cell. Phagocytosis. Think of phagocytosis as a cell eating larger particles. So larger molecules are going to be phagocytosed. Smaller things are going to be penocytosed. Think of penocytosed as a pinch. Smaller things are going to be penocytosed inside the cell. You have receptor-mediated endocytosis. Okay, exocytosis. So think of exit, transporting things outside of the cell. Okay, then transcytosis, you're transporting across, and then out of the cell. Okay, so you're going from inside the cell to across the cell, back out of the other side of the cell. Okay, you're going all the way across the cell essentially. And then vesicular trafficking, transport from one area of an organelle in the cell to another. So again, you might go from the Golgi apparatus to a lysosome. Okay, and so forth, and so on. Okay, but all these processes require energy. Okay. So endocytosis involves the formation of protein-coated vesicles. So usually involve receptors that can be very selective process. All right, some pathogens are capable of hijacking the receptors for transport to the cell. So I talked about that a little bit last time. So essentially, the receptors, the cell has naturally active receptors that it uses for endocytosis. Okay, that these are receptors that are naturally used for endocytosis. What happens is, again, viruses and bacteria, I should say, have been evolving with cells since the beginning of cells first were formed. Okay, they've been evolving with them. So they know that certain receptors that they can bind to triggers endocytosis. So what they do is they create antigens that can bind to these same receptors, and then they kind of trick the cell to endocytosis them with pathogens, and they end up, destroying the cell, making the cell sick, so on and so on, and making you sick. But essentially, they hijack the process of the normal endocytosis process of the cell. Okay. And once the vesicles are pulled inside the cell, a lot of times what they do is they fuse with a lysosome. Okay. And the lysosome, a lot of times it degrades things, and then it can recycle anything that can be recycled. And then if it doesn't fuse with a lysosome, then it's going to undergo transcytosis, meaning it's going to go across the cell and then be excytosed the other side of the cell. Okay. So it's showing you endocytosis. So you have your particles, and you have your pitted, coated pit that's going to ingest the substance. So essentially, it's going to form this this crevice that's dipping inside the cell, and it's going to pinch off. Essentially, it's going to come together, and it's going to pinch off and form this circular vesicle. All right, protein-coated vesicle. All right, that normally, a lot of times, is going to fuse with a lysosome. Okay, so it's going to uncoat itself. Essentially, the proteins are going to come off. That's what's uncoating itself. The proteins come off, and then it can fuse with a lysosome. Okay. And then a lot of times the lysosome is going to digest its contents and then deliver, or it can, if it digests its contents, then it can recycle things that it can recycle. But if it doesn't fuse with a lysosome, what can happen is it can just keep going to the other side of the cell and do what's called transcytosis and just be expelled out of the cell through exocytosis. Okay. And these transport vesicles, okay, they are going to go back. They are recycled, so they're going to go back to the plasma membrane for recycling them. Okay. All right, so phagocytosis, the way I remember it, I think about it as a cell eating. Okay, so a lot of times if you can watch a cell going through phagocytosis, there's many, I'm sure you can find it on YouTube videos. It shows, you see little projections, they call them pseudopods, that form around particles. Okay, that are going to be engulfed, and then they're going to form a vesicle and pull inside the cell. Okay, and the foreign vesicle is called a phagosome. Okay, and phagocytes digest foreign particles, anything that comes in foreign, that's in your blood, that's your macrophages and cover, could be. They're going to phagocytose. They're going to digest it so that it doesn't make you sick, essentially. All right, and phagocytic cells move by amoeboid motion, where the cytoplasm flows into temporary extensions that allow the cell to creep. Okay, and again, if you want to know what amoeboid motion is, just watch a YouTube video, and you can see one on YouTube very easily. So this is just a schematic showing you, again, phagocytosis. The cells engulf large particles, all right, and it does that forming these pseudopods, these false feet. It's going to surround. See how it kind of look like feet, or even maybe kind of like claws of hands, or even. that form around the particle, okay? And then when it encloses it, it's going to be called the vesicle is going to be called a phagosome. And a lot of times the phagosome is going to bind with a lysosome, okay? And undigested contents remain in the vesicle or are ejected by exocytosis. And the vesicle may things like macrophages binding to microorganisms like bacteria, which is going to possibly make you sick. Very important, all right, part of the immune system. So pinocytosis, you can think of it as the cell drinking. So these pino are going to be smaller things. They're going to be fluid phase endocytosis. So think of it as the cell drinking pinocytosis, okay? So the plasma membrane is going to enfold, bring in extracellular fluid and dissolve solutes inside the cell, all right? And this fuses with the endosome. And a lot of times the cell does this to sample the environment, okay? And this is also the main way that nutrients absorb inside the small intestine, okay? All right, so again, pinocytosis is showing you the cell drinking. All right, gulps a drop of extracellular fluid containing solutes into tiny vesicles. No receptors are used, so the process is non-specific, and most vesicles are protein coated. All right, so exocytosis, think of exit. All right, so it's a process where material is ejected from the cell. All right, it's usually going to be activated by cell surface signal, all right, or changes in membrane voltage. And the substance being ejected is enclosed in a secretory vesicle, all right, and the protein of the vesicle called V-snare finds and hooks up to the target. T-snare proteins on the membrane. Okay, so this, think of it as the vesicle has a protein called V-snare that's going to hook the targets, all right, which are T-snare proteins on the membrane, okay? So think of it as a vesicle coming in and it's going to use its V-snare to dock with T-snare, okay? All right, and that docking process is going to trigger exocytosis. And again, the cell does this for things that it wants to secrete outside of the cell all the time. Could be hormones, neurotransmitter, mucus, cellular waste, okay? So like I talked about previously, when a protein or a hormone or whatever has been synthesized inside of the cell goes through, you know, either the smooth ER or the rough ER, it goes to the Golgi apparatus, and then from the Golgi apparatus, it could be sent to the plasma membrane to be exocytosed, okay, in a vesicle, all right? So this is just showing you an image, okay? So again, on the vesicle, you have this V-snare, all right, protein that's going to dock, that's going to connect with the T-snare that's on the membrane, okay? So they're going to connect. That's going to trigger exocytosis, okay? So as soon as those V-snare binds to the T-snare, okay, that's going to trigger exocytosis. So the vesicle and the plasma membrane fuse and the pore opens up and the vesicle contents are released to the cell exterior, okay? All right, don't let this slide confuse you. So just summary of the passive processes. Again, don't let this slide confuse you, okay? Diffusion simple diffusion, facilitated diffusion, and osmosis, which is just the diffusion of water. All right, none of those processes require energy. They don't require ATP. Okay. Kinetic energy is just energy of motion. Okay. And anything that's moving has kinetic energy. Okay. So don't let that confuse you. Okay. So anything that moving has kinetic energy. But a lot of times what happens is stored potential energy gets converted to kinetic energy. Okay. The particles or water molecules, they're moving from high concentration to low concentration, and particles are always, there's always some type of vibration where particles are always moving. Okay. Essentially a solid, there's just very little movement. A liquid, they have more movement, they're more free to move. In a gas, there's a lot of movement in gas, but there's always movement in particles. Okay, they always are moving somewhat, even in solids. So just understand that. Okay, but don't let this slide confuse you. Passive transport does not require ATP. So we just say, we don't talk in terms of kinetic energy. We just say if it doesn't require ATP, then it doesn't require energy. Okay, so we don't talk in terms of kinetic energy. So don't let this slide confuse you. All right. So now we're going to get inside of the cell. Okay, and you know, and the cytoplasm is going to be surrounding the organelles within the cell also. Okay. All right, so the organelles that are in the cytoplasm are going to be the mitochondria. Think of, they mean little bodies. Okay. So think of how your body has organs in them, heart, kidney, lungs, and so forth. Okay, and all those organs work together so that your body functions properly. So think of organelles as analogy to like the organs in your body, and these organelles have to all work together so the cell can function properly. All right. All right. So the powerhouse of the cell is the it's the mitochondria, which generates ATP. The majority of it generates the majority of the ATP that your body uses. Okay, and that's through aerobic cellular respiration. Okay, and essentially The mitochondria goes through the Krebs cycle and the electron transport chain. And in the electron transport chain, oxygen is the final electron acceptor. That's why you need oxygen. And when oxygen accepts the final electron, it generates water. Okay. So again, mitochondria, very, very important. And the reason why I say generates the majority of the ATP, because there is what's called anaerobic meaning not requiring oxygen, cellular respiration, which is essentially glycolysis that happens in the cytoplasm. But glycolysis only generates 2 ATP, and that's not enough energy for large organisms. Okay, so really only certain bacteria can survive off of anaerobic respiration, okay, which is essentially just glycolysis, which they usually use the end product of glycolysis to generate lactic acid, ethanol, a lot of times what we call fermentation, okay. So again, higher level organisms, anaerobic respiration doesn't generate enough energy, so you have to go through the aerobic respiration, which involves the mitochondria. Okay, and just know that mitochondria has their own DNA, RNA, and ribosomes. Okay, so they resemble bacteria in a lot of ways. And just a side note, so you know how you have the evolution of cells. Essentially, what they think is that you start off with a simple cell, and then cells got more complicated by a larger cell engulfing a smaller cell, like a mitochondria, for instance, or like chloroplast, for instance, and then they ended up staying in a symbiotic relationship. So the mitochondria stayed inside the cell that engulfed it, and it became its own functional, organelle. So that's why it has its own DNA and RNA and ribosomes. Okay. So anyway, that's just kind of a side note. All right. So here's an image of the mitochondria. All right, and you see you have your inner mitochondria membrane, these cristae, which are used, these little folds, okay, are going to be used So these folds that are the site of protein synthesis, made up of protein and ribosomal RNA. So your rRNA, which organelle makes the rRNA? Okay, I talked about this in the first lecture. It's the nucleolus. The nucleolus, which is located in the nucleus, makes the rRNA. All right, so that rRNA is part of the ribosome. Okay, so that's going to be subunits that are used to make up the ribosome. Okay, so you have two types of well, I shouldn't say types, but you have two forms of ribosome found in the cell, either free ribosomes, which are freely float in the cytoplasm, okay, or you have the membrane bound bound ribosome, which are going to be attached to the endoplasmic reticulum. Okay. So again, just in general, your free ribosomes, those are going to synthesize proteins that are going to stay within the cell. So those are usually what the free ribosomes are going to be used for. Whereas your ribosomes bound to your endoplasmic reticulum, a lot of times those proteins made on the rough ER are going to end up being secreted outside of the cell a lot of times. And not always, but a lot of times that's the path that they follow. They go through the rough ER to the Golgi apparatus, and then they get intravesical secreted outside the cell. Okay. And again, you can also go from the Golgi to the lysosome, or you go from the Golgi to other organelles too. But again, for the proteins that get secreted, okay, they're gonna go through the rough ER, Golgi, and then to the secreted outside the cell. All right, from the Golgi vesicles leaving the Golgi apparatus. All right, so also remember that the endoplasmic reticulum is continuous with the outer nuclear membrane. And again, think of the endoplasmic reticulum as like a highway, okay? Think of it as the highway that's leading to the Golgi apparatus, okay? Think of it as a highway that's leading to the Golgi apparatus. And again, you got two varieties of endoplasmic reticulum. The rough ER, remember the rough ER is called rough because of the ribosomes attached to it. And again, proteins are made on the rough ER. They're made on the ribosomes on the rough ER. And the smooth ER has no ribosomes, and those are where lipids are made. Lipids are made on the smooth ER. And again, whether it's lipids on the smooth ER or proteins from the rough ER, they all go from the rough ER or the smooth ER to the Golgi apparatus. The Golgi apparatus is the post office of the cell. It's going to sort and package things and send them to their final destination of where they need to go. Okay. So here's just showing you an image of the endoplasmic reticulum. So again, it's continuous with the outer nuclear membrane, and you have the rough ER, have the ribosomes attached. Smooth ER, no ribosomes. Okay. All right. So again, rough ER. Already talked about this. So again, side of synthesis of proteins that will be secreted from the cell. All right, so again, think about the rough ER. You think about proteins that are going to be secreted from the cell. And it's going to go from the rough ER to the Golgi apparatus, where things are packaged and processed and then sent to the final destination. Okay. All right, so the smooth ER. Again, the smooth ER has no ribosome. So the smooth ER are going to make lipids essentially. Okay, so it's going to make lipids for lipoproteins. Okay, it's going to be involved in lipid metabolism. Be involved in cholesterol and steroid-based hormone synthesis. Okay, and also involved in, which we don't really talk about a lot, is detoxification of certain chemicals. Okay, and the storage and release of calcium, which is really important when we talk about the nervous system. All right, you talk a lot about the sarcoplasmic reticulum when we talk about the nervous system, and the sarcoplasmic reticulum is just a specialized smooth ER essentially found in skeletal and cardiac muscles. Okay. That store calcium. So specialized smooth ER that stores calcium in skeletal and cardiac muscle. We'll talk about that in greater detail when we talk about the nervous system. Okay. But these are the different functions of the smooth ER. So besides just making lipids, are also involved in detoxification of certain chemicals. And again, storage of calcium, and as the sarcoplasmic reticulum in the skeletal and cardiac muscles. Okay, but if you want to make things simple and just, if you don't remember anything else, remember they make lipids. Okay, remember that. But then when we get to the nervous system, we'll bring back up the sarcoplasmic reticulum. Okay, but just remember right now that they make lipids. All right, remember at least that much. All right, so the Golgi apparatus. So when you think about the Golgi apparatus, think of the post office. So the Golgi apparatus is like the post office of the cell. It's going to, so things that come from the smooth ER and the rough ER go to the Golgi apparatus, and then the Golgi apparatus is going to modify them and then package them to go to their final destination. Okay, so you have what's called the cis Golgi, which is the Golgi, the part of the Golgi apparatus that's facing the endoplasmic reticulum, and then you have the trans Golgi, which is the outer face of the Golgi that's facing like the plasma membrane. Okay, essentially what happens is the proteins or the lipids are going to fuse, gonna be transported to the Golgi in a transport vesicle. So it's gonna fuse on the cis side, okay, and then it's gonna be modified as it goes through the Golgi. Gonna be modified, tagged, sorted, and packaged. And then on the trans side, it's going to form another pinch off and form another transport vesicle that can go to like a lysosome or go to the plasma membrane, okay, depending on where the final destination is. And an image of it is going to make it more easier to see. So you have the cis right here, the cis side of the Golgi that's facing the ER. Okay, so the transport vesicles are going to fuse on the cis Golgi. It's going to go through the Golgi apparatus, get modified and packaged, and then it's going to leave on the trans side of the Golgi and transport vesicles, okay, that can be either secreted outside of the cell, or they might go to transport it to other organelles like the lysosome, okay. So this is just showing you again the pathway from the ER, endoplasmic reticulum. All right, vesicles pinch off of the ER. They fuse with the cis side of the Golgi apparatus, get packaged and modified, and then they bud off of the trans side. Vesicle buds off, and then it can go to different locations. It can go secreted outside the cell. It can go embedded in the cell, or it can go to a vesicle like the fused with the lysosome. Okay. So that's showing you the three pathways. All right. So peroxisomes are membranous sacs that contain powerful detoxifying substances that's going to neutralize toxins. So Um, one of the byproducts of using oxygen, so cells using oxygen, is the fact that oxygen creates free radicals, okay, which are very reactive and they are very damaging to the cell if they aren't contained and essentially broken down, okay? So these free radicals have to be neutralized. Essentially, the peroxisomes do that. They contain an enzyme called catalase, okay? An enzyme called catalase, which can break down hydrogen peroxide to oxygen and water. And then they also have oxidase, which is going to convert those free radicals to hydrogen peroxide. All right, so oxidase is going to first convert those free radicals to hydrogen peroxide, and then catalase is going to break down the hydrogen peroxide to oxygen and water. Okay, so again, it breaks down these free radicals in two steps. First, convert the free radicals using the oxidase to hydrogen peroxide, and then converting hydrogen peroxide to water and oxygen using catalase. Okay, those are two different enzymes. And peroxisomes also play a role in lipid metabolism. Okay, the breakdown and synthesis of fatty acid. So they also play a role in lipid metabolism. Okay. I don't remember it. All right, so when you think of lysosomes, think of Lysol. And what does Lysol do? It cleans. All right, lysosomes are the garbage disposals of the cell. Okay, so lysosomes have digestive enzymes that digest things. So it can digest bacteria. So remember things like remember how we talked about how bacteria and viruses can trick the cell to be in endocytose. So a lot of times those after something's endocytose, it's gonna fuse with the lysosomes. All right, and the lysosomes can digest hopefully lysosomes. Okay, but in the perfect world, they will be digested by the lysosome after they've been endocytosed. Okay, but the lysosomes also degrade non-functional organelles. Okay. The lysosomes also have metabolic functions. Okay, so they break down and release glycogen. They break down and release calcium from bone. And so intracellular release in injured cells cause them to digest. So apoptosis, I don't know if you heard of apoptosis, is like when cells are programmed cell death. So that's going to involve the lysosome when you have programmed cell death. So cells, after they are worn out essentially, and they know that they are no longer functional and they need to die, you can go through apoptosis, which is essentially programmed cell death, and that involves lysosomes. Okay, the intracellular release of injured cell cause cells to digest themselves, autolysis. Alright, so here's just a 10 or additional support, okay, inside the cell that helps reinforce the plasma membrane. Okay, so elaborate network of rods that run throughout the cytosol. So they kind of act like the bones, ligaments, and muscles of the cell. Okay, you have three main types, and they're classified based on how big they are. Microfilaments are the smallest, intermediate filaments are in between, and microtubules are the largest. All right. So microfilaments are made out of actin subunits. Okay, so they're the thinnest of all the cytoskeletal elements. So they are involved in cell motility and changes of the cell shape. Okay, also involved in endo and exocytosis. Okay. Intermediate filaments are in between. So their size is in between microfilaments and microtubules. All right, so they're tough and soluble rope-like protein fibers. All right, some have special names. Some are called neurofilaments and nerve cells, and keratin filaments and epithelial cells. Okay, we'll talk about keratin when we talk about skin. So keratin plays an important role, all right, in your skin. So we'll talk about that then. All right, but again, intermediate filaments are in between, as far as their size. They're in between microfilaments and microtubules. All right, so here's showing you the largest of the cytoskeleton elements, the microtubules. So most radiate from the centrosomes area of a cell. All right, so the microtubules are going to determine the overall shape of the cell. All right, and many of the organelles in the cell are going to be tethered to microtubules to keep the tubules as tracks. All right, to move things throughout the cell. All right, so we are going to actually, let's go ahead and do this slide too. So the centrosomes and centrioles. To the chromosomes, those spindles that attach to the chromosomes that move the chromosomes around during mitosis. All right, line them up in the middle of the cell during metaphase, separates them, sister chromatids during anaphase. So these centrioles, that's what they do. They make those mitotic spindles during mitosis. Very important. All right. So remember that. All right. So that is where we're going to stop for this lecture, part 2 of chapter 3. Okay, and